The overall goal of this procedure is to demonstrate the role of oxygen-glucose deprivation as an in vitro ischemia-reperfusion injury model for studying the blood-brain barrier dysfunction. This is accomplished by exposing brain endothelial cells grown in chamber slides to oxygen-glucose deprivation by replacing the media with deoxygenated no-glucose media and exposing them to a hypoxia chamber environment for 2 hours. The cells are then returned to normal conditions for an hour to achieve reoxygenation. After processing the cells, immunofluorescence staining of zonula occludens 1 and rhodamine phalloidin labeling of filamentous actin is performed. Cells are then imaged using a 60x water immersion lens on a confocal microscope to obtain single optical slices for analysis.
Demonstrating the procedure will be Himakarnika, a graduate student in my laboratory.
To begin the procedure, have primary culture RBMECs made from adult Sprague-Dawley rats on hand. Commercially available lines are also acceptable. Cultivate the RBMECs in 100 cm dishes coated with fibronectin. Culture them in rat brain endothelial cell growth medium and change the medium every other day. Grow the cells until they are 80 to 90% confluent. Then, gently wash the cells with 5 milliliters of PBS.